Okay. Hello, everyone.
Okay.
Um, good afternoon, good morning, and good evening. It's very honored to be here to present the research work from our group. My topic is about establishment of the resistance evaluation method of banana to Foc TR4 and the field trace. My topic includes three parts. First one is Foc in China. Second is resistance evaluation method. The last one is field trace and breeding activities. We all know about the Fusarium Wilt caused by the Fusarium sporin cubosense, this series fungi. So at the beginning of our research work, we collected lots of the samples from the five provinces in our country. We have the large scale of the producing areas in China mainland. Finally, we identified the 8 VCGs discovered in China. So this is the map of the Guangdong province. So we saw the red 5 stars. We also we can see lots of the TR4 fungi in across spread in this province. This is the Guangxi province. Actually, right now the TR4 fungi spread it all of the province, and also the Hainan province. We identify lots of the these TR4 fungi in, and the Fujian they also found lots of the fungi in this province. Actually, in Yunnan, we already have the TR4 fungi because they identify the collecting time is as early of the maybe 2011. So as last time we haven't found the TR4 fungi, but right now we have all of the fungi in TR4 fungi in Yunnan province. So then we developed the DGFP method to tracing the fungi, the TR4 fungi. They developed a resistance evaluation method. So from this slide, we can show the GFP fungi, the GFP fungi. We can show lots of the fungi how to invite the cell walls, and also the spores germinated in the cells. So these slides show how the fungi into the cells from the root and then in the cells of the cells tubes in the cells. So you can see also we can see here the spores germinated in the cells. It's a very serious problem. So we also found the lots of the difference between the resistance varieties and the sensitive varieties. We saw in these 3 resistance varieties, there are lots of the spores germinated. So the germinated is very higher than the resistance lines, the resistance varieties. And also we can see the effect of root exudate rates on spore germinated from above the 3 resistant varieties. We see very few or maybe less the spores, but we found lots of the sensitive varieties where we can see here lots of the spores germinated in the root zones and root area from the root systems. So, and we developed the resistance evaluation method in a greenhouse. This is the instance of disease in greenhouse. We can see lots of the symptoms with the different levels from 0 to 5. 0 is no symptoms, and the 5 is the very serious symptom in the rhizome areas. And also using the PCR detection from 0 to 5, if the symptom is very serious, so the band, the pattern is very stronger. So we will use this kind of PCR detection method. We can see the more serious and then the more stronger the band. From the field evaluation, we in different locations, we have several locations here. So from the field evaluation, we have the instance of the disease. We also, this picture shows the one location. We also have the very similar symptoms. From F, this is very clean and it's no symptom, and the K is very serious symptoms in rhizomes. We want to check this system is okay. We use 6 main cultivars as control. From the BASI is very sensitive, and the Zhongjiao number 3, Nomdao number 6 is a moderate resistance, and the Kanggu number 5 and the Zhongjiao number 4 is resistance clones. We will see this is called result. From this potting field, potting garden, we will see the BASI is a sensitive resistance. The sensitive varieties, we will see it should have some symptoms. The Zhongjiao number 3 is has a moderate varieties. We can see it looks healthy. So the potting, this is the potting area results. And then we use this system to evaluate the different kind of varieties. It includes Highland bananas and plantain. So this result of the Highland bananas and plantain, we will see the 7, it's 5 or 7, 5 Highland banana and 7 plantain. So it looks like the Highland banana and plantain are tolerant, or at least tolerant to TR4. This is quite good news for the countries who consume the Highland banana and plantain as their stable food. This slide just shows some Highland bananas. So you will see in the rhizome area, it's very clean, and also the plantain is very clean. So I think this this result is very similar to the results from the Indian scientists. Just Dr. Uma showed their result in the last presentation. It's very similar results. And also they evaluate the deployed banana germplasm. According to this list of the results, we will find almost the deployed are resistant to TR4, especially for the Pahang, the Kakita 4, and is a wild species. It's very high resistant to TR4. So this is also very similar work, like the Dr. Uma did in her presentation. So this slide show the deployed symptoms. You will see the rhizome area is very clean. So almost the deployed are resistant to TR4. And also they evaluate some of the other diploid and tetraploid varieties, like the small clone, and also the other AAB or AAAB varieties. Lots of lots of them are resistant to TR4. So these slides show our field work in the field. So we will see the field 25 is very highly resistant to TR4. It looks healthy. But for some of the varieties, like the field 17, Utasa, this is a Holland variety, and this is ABB variety. So it's the symptom looks very serious in the field. Maybe they are sensitive to TR4. So the above the results we published in several years ago, and one of the results published in recent years. For the breeding work, we are using the mutation work. Just Dr. Wuma also did the similar work. We use the chemical mutation using multiple bonds and embryo genetic colors. We use these the chemicals to induce the mutants or double the chromosomes. And we also use the physical mutation building technology to get the variation of types. But the work is very very hard for screening in the field. So we will see we use large scale of the field evaluation. So you will see this picture shows our Dr. Chunyi just standing with maybe a potential resistance lines or clones in the field. So this plant looks healthy, and you will see in other place in all the plants are infected by the fungi. So maybe these varieties, sorry, this is some clones are resistant to TR4. So using this system, we then the next step we using the rapid propagation to have a large scale of the plants in the field for evaluate. So using this system, we get some resistance clones or varieties. For example, this is the Zhongjiao number 3. Actually, this is Torrance varieties. It has good bunch and good taste, and the resistance is around maybe around 20 or 30. The taste is good. This is the Zhongjiao number 4. This clone is very high resistance to TR4. So in the field, the infection rate is it's just around 5%. So it has a good yield, and also the quality is excellent quality. But the problem is maybe is the cycle. I mean, the growing cycle is 1 month longer than the other varieties. This one is number 6. This is a moderate the resistance to TR4. It has good yield, also has good quality, and the cycle, the growing cycle is a little bit shorter than the Zhongjiao number 4. We have those clones in the field, so we want to large scale evaluation in the field. So this picture show our experimental field. So lots of the farmers to visit our the evaluation field. So we have Zhongjiao number 4 in different base, in demonstration base. In the this picture shows the city of in Guangdong, the Lianjiang city. So it seems like the Zhongjiao number 4 is good planting layer. So and also we are doing a crossbreeding. You know, crossbreeding has a long-term work. So we use prison work as the female parents. So actually, around the 10 years work, we finally get some tetraploid hybrids. This is just show our the potential varieties. It's a tetraploid, and it's the varieties has a very good bunch. The peel, I mean the color of the peel looks good, and the pump is the white. The taste is good. And we have evaluated this hybrids in the field for around 5 years. This variety, this hybrid is very highly resistant to TR4. Actually, we don't find any plant of these hybrids in the field infected by the Fusarium Wilt. So this could be a potential varieties, or maybe as the female parents for the secondary crossbreeding. For the molecular breeding, we use the agrobacterial material genetic transformation using a banana ACIAR. And recent years, we use the HIGAS. It means host-induced gene silencing. We use this kind of the technology to get the transgenetic plants in the field. It has the transgenetic lines has very high resistance to TR4 in the field. We published these results in the last year. We also using a genetic engine like the Professor James did in his lab. We did the very similar work using the CRISPR-Cas9. ULC also using this system. Actually, this system is very high efficiency in our group. So from the above the work, we hope we can use the breeding activities to overcome and finally to overcome these TR4 fungi, these serious problems. And I believe we can have beat these fungi. Thank you for listening to my presentation. Thank you very much.
Oh, thank you so much. Yeah, Dr. Ocean. Yeah, there are several questions for you. The first
Yes.
The first is the about the root exodes. What the method did you use to collect the root exodes? What concentrations of the root exodes did you add to the media to evaluate its effect on spore germination? I can answer this question for you, Hazel.
Okay. Yes, of course. It's your work.
Yeah, I
Okay.
collaborated with him about the. First, we cultivated different banana varieties in the liquid medium, and we determined the final concentration of the root exodes by the weight of the root system. We measured it about 10 grams 1 microliters. Then we used the primary concentration to treat the root exodes, treat the TR4 spores. That's okay, Dr. Yeah. Dr. Hazel. Okay, Hazel. Okay, then the second question is from Anne Bin. How long does it take for the hybridization of those species?
You mean the hybridization? I mean the crossbreeding? I'm not sure what's the meaning of these words. Maybe it's crossbreeding. Actually, for crossbreeding, we did from around 10 years right now. It's a really long time to get these results. but maybe it will cost more than 10 years or maybe 20 because it's a long way to go forward to this kind of breeding work.
Maybe the internet is not good. You can write in the chatting room. Yeah.
Yes. Yeah, I will put my email here so you can exchange with me using the email.
Well, the internet is not good. Yeah.
Yeah. Thank you.
Can you hear me?
Yeah, I can hear.
Okay. Okay. The third question is from Yak. He wants how many % of growers are using the Zhongjiao number 4, number 3? Are they yet to be commercialized?
Yeah, actually, Zhongjiao. Can you hear me? Can you hear me?
Yes, I
Sorry,
can hear you.
can you hear? Yeah, okay.
Yeah.
For Zhongjiao number 4, we also has the commercialized in the market. I'm not sure how many % of the growers, but it's around 100 hectares in the market, in the field, 100 of the hectares right now, because it's continue to be a demonstration in the field, in the market. Okay.
Okay. Yeah. Okay. The next question is from Selva Rajan. He wanted also has the question on the root exodes. Did you use the exodes collected at different stages of the plant growth and test against the TR4 spore germination? So what were the results? Okay, I can also answer this question. For the root exodes, we cultivated only the very small small plants, not very large ones. And for the a new paper will come out on the root exodes and the relationship with the root exodes to the spore concentration in the field. For the highly resistant, we also we invested the relationship with the exodes to the spore, the population size of the TR4 in the soil. For the highly resistant varieties, they really they keep the population size in the low, keep the pathogen in very low population size in the soil. And the new paper will come out in the next year. Okay, yeah. And the next question is from Uma. He wanted the Zhongjiao series of improved materials available for the public outside China. Is it in ITC? If not, when can we expect it to reach global programs?
Yeah, it's a very good question. Yes, it's not in ITC because we haven't sent to ITC. We hope we can exchange these materials with you, and we can talk in later in for future cooperation. Yes, I hope we can to reach the global programs because it has very good resistance to TR4. We want to evaluate in other countries. Yes, it's a good idea.